It's a genius in the lab with you.
Exactly.
Is OpenAI learning from this agentic AI?
Oh, yeah.
So there's a mutually beneficial relationship.
Yeah, I mean, we're not working with them directly on it.
But you use it, and because you
use
it with your AI,
right,
it's benefiting from it.
And we first thought to turn it into a company, because that's kind of one of the things we do in my lab, because I've always thought that it's important to give back to the taxpayer the money that they've invested in us. And the best way to do that is commercialization. I'm totally unapologetic about that, even though that got me in a lot of trouble at Stanford in the early days when, making money was, commercialization was evil, and even at Stanford. And so I think that that's an important process, because scientists are good at asking maybe the questions and coming up with solutions, but scientists aren't the best at commercializing it and turning it into a product that can be used, or testing it, in large communities. So the AI that we developed, we thought, okay, well, maybe we can do this. We thought, you know what? AI is moving so fast. Why don't we just give this to the community? Why don't we open source this? We can use it for maybe specific targeted purposes, but we're basically going to publish the whole thing on GitHub to let other people use it. Because we've seen other people make claims about stuff that they've already made, and it's like, ours is better. So why don't we just put it on GitHub and let people learn from it?
The resistance to the commercialization, what was the initial argument?
So back when I was a grad student in the 80s, basic research, as opposed to translational research, was considered the height of intellectual desire, right? Basic research, and we're not here to make money, we're here to discover things. And that's important. And nearly every major discovery and every major therapy in the world came from basic research. But then, there were limits to how much money you could give to basic research, and then there was a desire at a certain point to say, hey, are you going to do anything about this? Are you going to make it? And so translational research became a push. So there's a guy at Stanford by the name of Paul Berg, who won the Nobel Prize for recombinant DNA way back in the day, and Paul came up with this concept, bench to bedside, meaning that we don't have to be either or. We can be part of an arc, and Stanford wanted to be and enable within the medical school both the basic research, which we were great at, as well as bringing it directly to the patients as well. So to link clinicians and the desires of clinicians with the basic researchers. I mean, most scientists would be happy just to study anything. Just point me at something and I'll be happy if I can get interested in it. So, and we're no more happy than when somebody recognizes the value of what we do.
Right.
But basic research was sort of the height, and there was a push against anybody trying to commercialize. So when I started as an assistant professor, so I started as a grad student. I went to MIT to work with this guy, David Baltimore, who won the Nobel for reverse transcriptase. And then I wanted to come straight back to Stanford because I already felt that it was a positive environment for commercialization. My former boss's mentors, Len and Lee Herzenberg, had two of the biggest patents at Stanford. They had the fluorescence activated cell sorter and then what are called humanized antibodies, which brought in hundreds and hundreds of millions of dollars to Stanford. And actually, they gave personally most of their own money away. They kept enough to survive, but then they gave most of the money away and they ran their own lab off of a lot of that money. But so I had learned from them about how to still do basic research but commercialize on the side. And so I wanted to bring that back, but the department that I came into, the Department of Pharmacology at the time, I was warned by many professors, don't commercialize that. And I ignored them, and I went and started a company that went public on NASDAQ. And many of those same professors came back to me, years later and sitting in my office asking me how to start a company.
Why did you, was it just a courageous decision to ignore them? Was it instinctual?
It just was instinctual. It was because I couldn't see the NIH funding what I wanted to do. Mm-hmm. So I had developed a way, this will sound scary, but I had developed a way to use retroviruses and make libraries of retroviruses to reverse the process of evolution in a way that rather than viruses hurting the cell, I set it up so that viruses would help the cell. And once they helped the cell, I would figure out what they did. And so we sold hundreds of millions of dollars of targets that way, using retroviral libraries to basically find targets and use some of the benefits of viruses, but to our advantage.
Just the concept of reversing evolution is fascinating, because it comes with, there's so many ethical implications, but if you didn't have any of those,
Yeah.
and you could do that large scale.
Well, I had developed in David's lab, along with this guy Warren Pear, a means, it's called the 293T retroviral producer system. It was a way to make large numbers of these viruses very quickly. It really followed on the work of this guy, Richard Mulligan, who'd also been a postdoc with David Baltimore, who developed what was called the 3T3-based retroviral production system. And he developed it in Paul Berg's lab at Stanford. So there's a lot of sort of, interbreeding here. But the problem with that was it took 3 months. So I had brought with me a cell line called 293T that I introduced to the lab and said, hey, maybe we could use this to make viruses quickly. I won't go into the details of why, but we could do it in 3 days rather than 3 months. And so that now, tens of thousands of labs use that worldwide, and it probably generates the most money for me every year over any of my other inventions. Just because Stanford, rather than patenting it, licenses it. And licenses are forever, whereas patents have a 17-year lifespan. So Stanford made a good choice there.
So do you think it was just a bias, an academic bias, like we shouldn't be focusing on money, we should be focusing on the work?
Yes.
And they missed the forest for the trees?
Right. But then people, they eventually learned,
Right.
and I wouldn't say that it's the way that people think anymore. But there's still a little bit of you shouldn't walk into the lab thinking I'm here to make money.
That's what they're worried about.
Yeah.
Right?
Right.
They're worried about the bastardization of it all.
Right. And so Stanford in the early days set up very clear lines about once you start a company and you license the patent or the idea to the company, you can still be involved with the company, but there's not a pipeline of technology now from your laboratory to that company. So they set up, an oversight board for each of these licenses that makes sure that, the students are not being abused. Because you don't want students, you don't want to be, covertly getting your students to do something that then you're going to walk behind a back door and then hand over to a company.
And patent it.
Yeah. It's so interesting that there's often very much a lot of worry that that's going to happen. But frankly, more often is the case that the company doesn't need the inventor anymore. In fact, I can't tell you the number of times that once the company's set up, they want nothing more to do with me because they have their own thing to do. They don't want the crazy academic coming in and vetoing their ideas. There's places for that where people like, Steve Jobs needs to hold on to the image of what he wants the company to be, as opposed to, I would probably be fired from a company within a week because I just don't like people telling me what to do. That's just a fact.
Yeah. So where you're at right now with this cancer research, when will this be applied in real world scenarios?
It already is.
It is.
Already is. Look at who just won the Nobel Prize last year, David Baker at Google, with the, ability to predict protein structure, et cetera. And protein structure, once you know the protein structure, now you can predict molecules that might come into it. So go back to the stuff that I'm trying to do with looking at the complexities of the dance of how the immune system talks or doesn't to cancer. If we can find a particular place that might be an Achilles heel along the way towards the shutting down that is different, for instance, than what the current drugs are, well, maybe we should aim at that. There's so many more opportunities that are suddenly opening up in front of us because the AI and the data is letting us look at a network of how the system is working. Before, it used to be you'd look at a computer chip and you'd see just a computer chip with a few wires. But imagine now that you, as a scientist, have a microscope that's looking at the complexities of the wiring diagram that's connecting this resistor to that capacitor to that diode to this transistor. That's where we are now. And so now suddenly we can say, well, I don't want to do that because it'll kill the chip. But the chip is malfunctioning, so let me put a little bit of pressure there, and now I can reactivate the immune system or the chip to work in the right way again.
So when